main objective of the laboratory was to extract DNA from a plant, with the necessary care to prevent the sample from becoming contaminated. A commercial kit was used and the CTAB protocol was followed, which was modified for the extraction of aromatic plants. In our practice, we used Agastache mexicano, previously ground and dried.
Materials and equipment. Extraction buffer, chloroform isoamyl alcohol, 5 molar sodium chloride, 3 molar sodium acetate, ethanol 70%, ethanol 95%, lysis buffer 250 milliliters, TE buffer, cold isopropanol, plastic tubes 2 milliliters, centrifuge for 2 milliliter tubes, water bath.
We had to mark 2 tubes for the respective samples. We also had to prepare our micropipette tips.
We need to weigh 20 milligrams of dry plant tissue. After that, we need to add 600 microliters of extraction buffer and mix the solution with a vortex.
Before we can centrifuge the 2 tubes, we need to balance them in weight so we can avoid spillage inside the microfuge.
The third step is to centrifuge at 4 degrees for 10 minutes at 11,600 revolutions per minute. When the time passes, we need to remove the supernatant and add 400 microliters of lysis buffer. After adding the buffer, we need to incubate the samples at 65 degrees for 30 minutes and mix the solution every 10 minutes. After the 30 minutes, we need to transfer the samples to ice and add 500 microliters of 24:1 chloroform isoamyl alcohol and mix well by immersion.
In addition to the practice, we had to prepare 1 liter of ethylic alcohol at 96%. The tricky part was that we had to prepare this 1 liter from a container with ethylic alcohol 70%.
Centrifuge at 4 degrees Celsius for 10 minutes. You should have the 3 layers top, aqueous phase, middle, debris and proteins bottom. Pipe it off the aqueous phase, taking care not to soak up any of the middle or chloroform phases. Place the aqueous phase into a new label plastic tube. Estimate the volume of the aqueous phase. Measure this new volume. Add the same quantity 24:1 CIA as the recovered volume of the supernatant. Centrifuge at 4 degrees Celsius for 5 minutes. Centrifuge at 4 degrees Celsius for 5 minutes at 8700 RPM.
In the first session of this practice, we accidentally threw away the aqueous phase of one of the final steps. So we had to remake again the previous steps, which include adding chloroform isomeric alcohol and centrifuging the samples. Despite the backdraws, we could finally put our samples in the incubator to finalizing the reactions.